Light-dependent systems. A photon of light is captured by photosystem II's antenna. Energy transfers electrons to the reaction center of photosystem II. Energy splits molecules of water into hydrogen ions and oxygen. Oxygen is released into the environment, which we use to breathe through cellular respiration. Hydrogen ions and electrons are carried by electron carriers. Hydrogen is released into the cycloid lumen before it reaches photosystem I.
A photon of light is captured by the antenna in photosystem I. Energy from the photon of light transfers electrons to the reaction center of photosystem I. Electrons pair with the hydrogen ions, which allow it to combine with NADP in the stroma. This forms NADPH, which is then used in the Calvin cycle. The Calvin cycle is a light-independent cycle within the process of photosynthesis. In the first step of the carbon cycle, carbon fixation occurs. In this step, carbon dioxide molecules combine with ribulose biphosphate within the enzyme Rubisco. This combination forms an unstable 6-carbon molecule. The unstable molecule splits into 2 stable molecules of 3-PGA. These 2 molecules of 3-PGA are activated by 6 molecules of ATP. 6 molecules of ADP are then released.
NADPH reduces to form 2 molecules of G3P. 2 G3P molecules may leave the cycle to form glucose. If the G3P remains, it may use ATP to regenerate into ribulose biphosphate. This releases ADP, which is used in chemiosmosis. Chemiosmosis is another light-independent process. There's a high concentration of hydrogen ions within the cycloid lumen. Hydrogen ions travel to a lower concentration on the stromal side through the ATP synthase.
The passing through of hydrogen ions causes the ATP synthase to rotate. As the ATP synthase rotates, ADP combines with phosphate. This combination forms ATP, which will be used in the Calvin cycle.
Cellular respiration begins through an anaerobic process, which means the process is done without the use of oxygen, and it's called glycolysis, which is found in the cytoplasm of all cells. Due to glycolysis, 2 ATP molecules transfer energy into the glucose molecule, creating 6 carbon sugar diphosphate molecules.
However, when the molecule splits, it creates 4 ATPs, 2 pyruvates, and 2 NDHs. When oxygen is added in this process, it starts the second step of cellular respiration, in which the pyruvate and NDH move to the mitochondria. When the pyruvate enters the mitochondria, it forms into an acetyl-CoA, and shortly after that, the process of oxidation begins. In oxidation process, NADH is formed, and due to the loss of carbon, carbon dioxide is formed. And during this process, it is being prepared to enter the Krebs cycle.
The Krebs cycle, also referred to as the citric acid cycle or the tricarboxylic acid cycle. In the Krebs cycle, pyruvate is used to make acetyl-CoA, and the cycle transforms acetyl-CoA into NADH, FADH2, and ATP.
In the Krebs cycle, acetyl-CoA is used to pick up groups of carbon, and in doing so, it releases energy in the form of reduced NADH and FADH2. During a complete cycle, 2 carbon groups are added, and because of carbon oxidation, carbon dioxide is formed. For each glucose molecule, 8 NADHs, 2 FADH2, 2 ATP, and 6 CO2 are produced.
Electron transport chain. Electrons get transferred between the membrane protein. The cell uses its energy to create ATP molecules.
Chemiosmosis. In the process of chemiosmosis, electrons have the ability to transfer hydronium ions through an ATP synthase. As it comes to the other side of the membrane, it gives the ADP energy to attach itself to another phosphate, thus creating ATP. In conclusion, 1 glucose creates 36 ATP molecules.
And now we'd like to play a little song we found known as the Krebs Cycle Song by none other than the Science Music Videos channel.
matrix of the mitochondria of all our cells is the cycle of reactions that won Hans Krebs a Nobel Prize. The cycle takes the energy in food and makes it into other forms your cells can use. Krebs cycle makes electron carrier NADH, which later brings electrons to the electron transport chain. And Krebs makes FADH2, its function is the same. Krebs also makes some ATP, another claim to fame. We're talking Krebs. It's a citric acid cycle, Krebs. Tricarboxylic acid cycle, Krebs. Each cycle makes one ATP, 3 NADH, 1 FADH2. Right before the cycle's a transitional part, links Krebs to glycolysis so Krebs can start. Enzymes break a CO2 off a pyruvate, yields an NADH and acetyl-CoA. Acetyl-CoA carries carbons too, with all the energy that derives from food.